Construct 1, Item 1. Following a dispute over the biological children of the late Paul Kafeero, forensic scientists conducted DNA paternity analysis using the polymerase chain reaction. During laboratory analysis, preservation chemicals disrupted the phospholipid bilayer of plasma membranes, reducing the activity of carrier proteins responsible for transporting nucleotides and other PCR reagents into cells. In addition, a laboratory contaminant inhibited cytochrome C oxidase in the electron transport chain, reducing ATP production required for active transport and other cellular processes. Some PCR reaction mixtures also contained compounds that inhibited Taq DNA polymerase, resulting in incomplete amplification of target DNA fragments. Despite these conditions, some cells remained viable by activating physiological survival responses. Let us analyze the table. The table shows cellular responses for survival. Pause and analyze. Task Part A. Analyze how membrane transport, ATP production, and PCR DNA amplification contributed to the low success of DNA paternity determination for Paul Kafeero. Part B. Evaluate the effectiveness of the observed cellular survival responses in enabling cells to survive under the laboratory conditions. Analysis. As usual, what happened, why it happened, and how. Do not forget to get self-study A-level biology textbook from all bookshops in Uganda. Membrane transport. Membrane transport was reduced because preservation chemicals disrupted the phospholipid bilayer of plasma membranes. This altered membrane fluidity and reduced the activity of carrier proteins responsible for transporting nucleotides and other PCR reagents into the cells. Consequently, insufficient PCR substrates entered the cells, reducing the amount of DNA prepared for amplification and contributing to the low success of Paul Kafeero's DNA paternity determination. Greetings to the students of Kimaanya Masaka Blessed Sacrament, Soroti Secondary School, and Mbale Secondary School. ATP production. ATP production decreased because a laboratory contaminant inhibited cytochrome C oxidase of the electron transport chain. This prevented efficient transfer of electrons to oxygen, reducing proton pumping across the inner mitochondrial membrane and lowering ATP synthesis by ATP synthase. Consequently, ATP-dependent processes such as active transport of PCR reagents and other cellular activities were impaired, reducing the efficiency of DNA sample preparation and PCR analysis. PCR DNA amplification. PCR DNA amplification was reduced because inhibitory compounds blocked the activity of Taq DNA polymerase. The enzyme therefore failed to efficiently extend primers by adding complementary nucleotides to the growing DNA strands during each PCR cycle. Consequently, only small amounts of the target DNA fragments were amplified, producing insufficient DNA for accurate comparison between Paul Kafeero's DNA and that of the claimants, thereby lowering the success of paternity determination. Evaluation. As usual, mechanism and effectiveness, limitations, and explained recommendation. Anaerobic respiration was effective because the proportion of cells using it increased from 5 to 62%, allowing glycolysis to produce small amounts of ATP despite inhibition of aerobic respiration. This ATP maintained essential cellular processes, enabling some cells to survive. However, anaerobic respiration produces only 2 ATP molecules per glucose and causes lactic acid accumulation, lowering cellular pH and impairing enzyme activity. Consequently, ATP was insufficient to maintain normal cellular functions, contributing to the decline in cell survival from 99 to 68%. Recommendation. Remove the inhibitor of cytochrome C oxidase and use fresh, uncontaminated culture medium before sample preparation. This restores aerobic respiration and oxidative phosphorylation, producing sufficient ATP for active transport, membrane repair, and other energy-dependent processes, thereby improving cell survival and DNA preparation. Expression of heat shock proteins. Expression of heat shock proteins was effective because it increased from 8 to 71%. These proteins acted as molecular chaperones by preventing protein denaturation and refolding damaged proteins, maintaining enzyme activity and improving cell survival. However, heat shock proteins cannot repair damaged plasma membranes or remove inhibitors of Taq DNA polymerase and cytochrome C oxidase. Consequently, PCR efficiency and cellular metabolism remained impaired. Recommendation: purify DNA samples and PCR reagents before PCR to remove inhibitory compounds. This restores the activity of Taq DNA polymerase and respiratory enzymes, improving DNA amplification, cellular metabolism, and reducing cellular stress. Increased membrane lipid synthesis. Increased membrane lipid synthesis was effective because it rose from 12 to 66%. Newly synthesized phospholipids repaired damaged plasma membranes, restoring membrane integrity and improving carrier protein-mediated transport of PCR reagents into cells. However, membrane lipid synthesis requires large amounts of ATP. Yet ATP production remained low because cytochrome C oxidase was inhibited. Consequently, membrane repair remained incomplete, reducing reagent transport and overall cell survival. Recommendation: remove preservation chemical residues and prepare fresh viable cells before DNA extraction. This preserves membrane integrity and carrier protein function, improving transport of PCR reagents, reducing membrane damage, and increasing the success of DNA paternity determination. Thank you for watching.